a concept that's called gene linkage. Probably want to see what I'm writing, huh? Okay, gene linkage. So, gene linkage basically says that there's this concept of linked genes. And we can say that linked genes are genes that are located on the same chromosome. That are inherited together more than 50% of the time. Okay? We say that non-linked genes are expected to be inherited together 50% of the time. Okay, so let's look at eye color and hair color. Okay, so if we were in a situation where they were linked genes, we'll do linked genes over here, and over here will be unlinked genes, non-linked genes. Okay, so we're going to draw the diploid cell. And just for the ease of drawing, we are going to draw it before it's gone through S phase. So each of the chromosomes is just going to be one thing, not the two things, right? Okay, so in the case of non-linked genes, you've got a chromosome from the father, and then this is going to be another chromosome. It's going to be a slightly different size, right? You can see this one's smaller down here, right? I guess we could say that one's from the mother because it's red. This one's going to be from the father. Okay, and let's say that this individual here is going to have the blue one. Let's say the allele on the blue one is going to be hair color brown. Okay, and the allele on the little chromosome for the blue one is going to be eye color brown. Okay, then on the same gene locus here, we could have a different allele. So let's say this one is hair color blonde. Okay, and again the same gene locus, so roughly the same place on this other chromosome here. We've got eye color blue. Okay, so this person, if this is a diploid cell from somebody, this person here has what phenotype? What would you expect this person to look like? They would have brown hair and brown eyes because they have one copy of the brown hair allele and one copy of the blonde hair allele, and brown is dominant to blonde, and so that means that they would show this brown, and the same thing with eyes, brown eyes and blue eyes. Brown is dominant, so it'll show brown. Right? Everybody following so far? All right. So in this case, this person could make some different gametes, right? So the gametes that can be produced by this individual are brown hair, blue eyes, right? So these are the gametes possible. You've got brown hair, blue eyes. You've got brown hair, brown eyes. You've got blonde hair, brown eyes. And you've got blonde hair, blue eyes. Right? Does everybody see what I did here? I just combined all the different things. I said, okay, this could go with this could go with that could go with that could go with that. Right? Yes? What's the law that says that this is going to happen, that these are going to segregate so that you don't have a brown hair and a blonde hair going to the same side? Segregation, yes, and then specific to meiosis, independent assortment, right? Okay, now, let's say in this left side here that these are linked genes, okay? So we only need to have one chromosome drawn. Okay, and so let's say this gene locus up here has hair, HBR, and this gene locus down here has EBR, or big EBR. Okay, and this one over here has the same gene locus for little h, little b, and this gene locus down here is for little e, little b. So for this one, the gamete's possible because of the law of independent assortment. Which possible gametes do I have? Look, these are on the same chromosome, right? So this chromosome is going to get pulled this way, this chromosome is going to get pulled this way. That means that your only two possible gametes are big HBR, big EBR, or little HB, little EB. Make sense? So, in this case, you'd have to run an 8x8 Punnett square to figure out the probability of what the offspring would be. But in this case, you only have two possibilities, and therefore only a 2x2 Punnett square. Or this would be a 4x4 rather. This would be a 2x2. Right? Okay, so let's do that then. Let's say here's our non-linked example. Okay, our non-linked example is going to have a male that is brown hair and brown eyes. That's going to be this example over here. So he's going to have HBR, HB, and then EBR, EB. So big HBR, HB, and then big EBR, little EB. Okay. Our female, though, is going to be blonde hair and blue eyes. And so those are both recessive, so we know that she just has to be all recessives. So again, to figure out the gametes that could be in this, you could do like you do in math, where you FOIL it. You do first, right? So this one, and then outside, so this one, and then inside, so this one, and then last, so those two. And those will give you all the possible gametic combinations, right? But then you have to draw a 4x4 Punnett square, and that sucks because that's a lot of stuff to have to draw. So the easier way to do this is to do two 2x2 Punnett squares. So we're going to do a Punnett square here, Punnett square here. Okay, this Punnett square is going to be for hair, and this one's going to be for eyes. So for hair color, you've got the male that has H or big HBR and then little HB, and then the female that has little HB, little HB. Big HBR, little HB. Little HB, little HB, and then big HBR, little HB. Little HB, little HB. Over here, oops, wait, no, we're okay. Little over here we got big EBR, and then little EB, and then little EB, little EB. I think the book made some reference that like you're supposed to always have the male on the top and the female on the side or something like that. It doesn't matter at all. It's totally inconsequential. Okay, so in this one we got big EBR, little EB, and then down here big EBR, little EB. Little EB, little EB, little EB, little EB. So, what outcomes do I have down here for hair color? What two phenotypes? What's this phenotype here? Brown hair. So 50% of the time, we're going to have brown hair offspring, and 50% of the time, then, we'll have blonde hair offspring. And then over here, we got 50% brown eyes and 50%. That was ugly. 50% blue eyes. So now is where the math comes in that saves you from having to do a giant Punnett square. What are the odds that offspring will have brown hair and brown eyes? 50% times 50%, which is 50% of 50% is 25%, right? So you just multiply. You do 50% times 50%, you get 25%. You can do the same thing here. So basically, we're going to figure out here that offspring for this cross could be probability. It's going to be 25% brown, brown. 25% brown, blonde. Nope, that's ridiculous. I mean, I guess it's not ridiculous, but I was doing eye color second, so brown, blue. 25% blonde, brown, and then 25% blonde, blue. All right. So we say that each of those is inherited together 50% of the time, right? 50% of the time there's brown and brown. 50% of the time there's not brown and brown, right? So 50% of the time you have two instances here where you have brown. 50% of the time it's with brown. 50% of the time it's with blue, right? Yes? Okay. So what about linkage? Linked example. Same exact parents. So the male is going to be brown, brown. And he's going to have the same phenotype as before, so that, or same genotype rather as before. So that's hair, big HBR, little HB, and then brown eyes is big EBR, little EB. Okay, and then the female was blonde hair, blue eyes, And so she's got little hb, little hb, little eb, little eb. Okay? So, to do this, we just need one Punnett square. Okay? Because, as we said, these genes are linked together. When we drew the gametes up here, or when we drew the diploid cells rather, we said that either this chromosome could go over here or this chromosome could go over here. But there's no ability for the little hb to be with the big E BR, right? So we know that the possible combinations up here are big H BR, big E BR, right? And then little HB, little EB. Okay, and then over here you've got little HB, little EB, and then little HB, little EB. So we got big HBR, little HB, big EBR, little EB, big HBR, little HB, and then big EBR, little EB, and then over here we got little HB, little HB, little EB, little EB, and then little HB, little HB, little EB, little EB. So what offspring do we have here? I'll let you finish your drawing and then we'll talk about it. Okay, so what are my two phenotypic combinations for my offspring? Yes, these two over here are brown hair, brown eyes. These two are blonde hair, blue eyes. So we're 50% brown hair, brown eyes. 50% blonde hair, blue eyes. Okay. What if we saw this? What if we were looking at real data and we saw this? How's that even possible? Have you ever seen somebody with brown hair and blue eyes before? Yes. Have you ever seen somebody with blonde hair and brown eyes before? Yeah. Are they as common? No. They're not nearly as common as blonde hair, blue eyes, or brown hair, brown eyes, but they happen. So how does it happen? What do you think? Nope. Simpler than that. More common. Something that happens all the time in meiosis. Not non-disjunction, nope. Crossing over, right? So let's say we've got this here, okay? And this chromosome crosses over this one, but it only crosses over at the bottom. Let's say the top part, I know I can't like rotate or anything, but let's say it only crosses over on the bottom part. It swaps then the EBR for the EB, right? And so now you've got two different gene linkages. That's why if you see somebody who has brown hair and blue eyes, generally they also have a parent who has brown hair and blue eyes, right? Because that crossing over happens some other time, right? So now that creates a new linkage, and so all the way through their bloodline then, they're going to have brown hair and blue eyes as linked, right? Until a crossing over happens again. Make sense? Okay. What if the genes are located right next to each other? What if instead of being at the top of the chromosome and the bottom of the chromosome, they were like literally right next to each other like that? What does that do? if crossing over, then if they were right next to each other like this and crossing over happened, right, what would happen? They'd both cross over, right? And so it wouldn't change anything. And so in cases like that, we would say that even though crossing over occurred, it didn't change the frequency in which they were related, right? And so we can say that if we know the percentage of time that two genes are being inherited together, we can tell how close together they are on the chromosome, right? The more often they're inherited together, the closer they are. Yep.
So does that mean that that's why people with red hair tend to have freckles? Are they close to each other?
Yep. Okay, we can say that the distance of two linked gene loci is inversely, or the distance between, two linked gene loci is inversely related to the percentage of time they are inherited together. Okay? In other words, if they're right next to each other, they're inherited together all the time. Right? So zero distance would be 100% of the time inherited together. The more the distance goes up, the less they're inherited together, because crossing over is more likely to happen on one of them and not the other one at the same time. Make sense? Okay. Let's talk about what tonight's actual lesson is about, which is on the genetic material. So I'll come right out and say it. DNA is the genetic material. You guys know that, right? DNA is the genetic material. Did you know, though, that we actually have not known that for very long, right? Less than 100 years we have known that DNA is the genetic material. In fact, for a long time before that, we thought that proteins were the genetic material. And the reason why we thought that was fairly solid in its science, and that was, they said, hey, we know that DNA is comprised of just 4 different molecules. There's only 4 nucleotides that make up DNA. And proteins have 20 different amino acids. And so if we see how different everything is, if we see that plants are really different from humans and bacteria is really different from plants and so on and so forth, that it's unlikely that the 4 different nucleotides in DNA are going to be able to cause all those differences, right? Turns out they were wrong about that, right? But it made some sense, some logical sense to think, okay, everything's very different, so we're going to need a whole bunch of different types of molecules, like 20 different amino acids, in order to get all these differences coded for. So how do we figure out that DNA is, in fact, the genetic material? Well, and so I guess we can put on here that 4 nucleotides account for all the differences in living things. Okay, so there's just A, C, G, and T, and when you put them together in different combinations, you get all kinds of different crazy living things, right? Okay, so the experiments that were done in order to determine that DNA is the genetic material are really easy to understand. They're really simple experiments that had really profound results. Okay, so these are the experiments to determine genetic material. So back in the 1920s, we figured out that bacteria have this really crazy thing that they do called bacterial transformation. And basically what happens in bacterial transformation is that bacteria can take in random DNA, right, and start expressing it. Okay, so bacterial transformation said that bacteria and take in DNA from the environment.
What do you mean by take
They like endocytosis it and bring it into the cell, and then they just like start doing it. Like, hey, cool, new instructions for something. Let's try it out and see what it makes. Right? Sometimes it works out in bacteria's favor and they make, you know, some awesome new novel protein that helps them. Sometimes it's super bad and they make a protein that kills them. But the thing about bacteria is like they're expendable. They reproduce so quickly that it might as well, it's in their benefit to just try it out. Okay? So they take in DNA from their environment, start expressing it, and transform their phenotype. It's back in the 1920s that we noticed this. And so in order to kind of to work with it, the easiest thing that you can look at is slime coats. So under a microscope, it's really obvious if a bacteria has a slime coat or not. I don't know if you remember back when we talked about prokaryotes, there was this layer of sugars on the outside of a prokaryote called a glycocalyx. And if it's organized, that's called a capsule. Right, and if it's disorganized, that's called a slime coat. And the disorganized ones, it gets really like hyperhydrated, they call it, and so it looks just like snot around the bacterial colony, right? So it's really easy to see on a petri dish. So let's say we take two bacteria. Okay, so the experiment worked like this. Okay, you have bacterial strain A. Bacteria A. Let's say that bacteria A produces a slime coat. And bacteria B has a capsule. Okay? So if you take bacteria A and you kill it, okay? You kill it by whatever means you want. You can boil it. You can expose it to some chemicals that will destroy it. You can starve it, whatever you want, right? You can blowtorch it a little bit. Point is, you just want to kill it. You want to make sure that that bacteria is totally dead. Okay? They're going to take that bacteria, throw it into a little blender, blend it up a bit, right? And then they're going to pour that bacteria onto the other one. Okay? So if you take bacteria A, so you kill bacteria A, you add to live B bacteria. The result of that is that you end up with about half of the bacteria that have slime coats and half of them that have capsules. So both slime coats and capsules produced. Okay, this was like the baseline experiment where they're like, hey, look, we can take this dead bacteria that produced a slime coat and pour it onto this living bacteria, and the living bacteria will start to make the slime coat based on taking in that DNA. Okay? So, they're like, okay, well, we can easily prove then that proteins are the way that genetic code is transferred. So they kill A, they mix with proteases, add to live B. And they were like, we killed all the proteins. We digested them with enzymes. And so we know what's going to happen. Of course they're not going to transform. And what they found was both slime coats and capsules produced. They always say that the best thing that can happen in a science lab is that you end up with results that you're not expecting. Okay, and that's what happened there. They were like, what? That shouldn't have happened. So they repeated it a bunch of times, and sure enough, every time they were transformed. So they're like, okay, let's try out the rest of the things. Let's kill A. Let's mix with RNase. Add to live B. And we get both slime coats and capsules. And they're like, well, it's not RNA then. And they kill A, mix with DNA ACE, add to live B. Only capsules produced. Which means no transformation. This was a serious eureka moment here, okay? Because this did not cause a transformation, that disproved what people had thought for hundreds of years, and that, not hundreds, but about 100 years before that, and that was that DNA was the heritable, or that proteins were the heritable information and DNA was not. Okay, and this one little experiment here kind of turned that on its head and said, look, when we kill the DNA, when we destroy the DNA with DNA aces, that prevents the transformation from happening. But if we don't do that, if we just destroy the proteins, then the transformation happens anyways, which means that the information to make this other stuff is not on the proteins, it's in fact on the DNA. Right? Questions over that? All right, that's it.